Armando Hasurugan, biology and medicine videos. Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook Armando Hasurugan. Please like, and here you can also ask questions, answer questions, and post some interesting things, including artwork, so you can send it to me. And you can also change the quality settings to the highest one for better graphics. In this video, we're going to talk about bacteria, and it's just going to look at the structure specifically. So, bacteria. Bacteria are the largest group of microorganisms of medical significance. And so, that's pretty important to know because they are literally everywhere. Bacteria occur in three main shapes: a coccus, a circular shape, sphere, a bacillus, a rod-shaped, and a spiral. Now, there's also other different shapes bacteria can take, such as a coccobacillus, which is in between a coccus and a bacillus. And there's also a bacteria can take shape of what's called a vibrio, and it's sort of spiral and bacillus in between. It's sort of got a bend to it. Now, some bacteria are surrounded by polysaccharides containing capsule, so a capsule containing polysaccharides. Now, this capsule, also known as a slime layer, actually allows the bacteria to bind to surfaces of different cells in our body, for example, and also to evade phagocytosis, to avoid phagocytosis. So it's very important, essentially, for the bacteria's survival. So what other parts does a bacteria have? What other structures do a bacteria have? Here I'm drawing a rod-shaped bacteria. It can be any type of rod-shaped bacteria. And now let's learn about part of its anatomy. Some bacteria have what's known as a flagella, which are thin, rigid filaments, and these allow for bacterial movement. Flagella are essentially made up of proteins, and these proteins can move, can propel the bacteria forward or move it around. Bacteria can also possess hair-like appendages coming out of their surface. There are two types of hair appendages, fimbriae and pili, and these are used interchangeably. A pili is typically shorter than a fimbriae, but they essentially do the same thing. The fimbriae contributes to the bacteria's ability to cause a disease by binding onto a surface of a cell in our body, for example. And this can also be done by the pili. The pili is another hair-like appendage, just like the fimbriae. And the pili help attach the bacteria to mucosal cell surfaces. And the pili are also involved in what's called bacterial conjugation. Bacterial conjugation is essentially the transfer of the genetic material of a bacteria from one bacteria to another by forming a bridge using a pili. Now let's look at the different layers a bacteria has. The bacteria's outermost layer is known as a capsule, and as we know, it has polysaccharides, which enables it to bind to cell surfaces and also avoid phagocytosis, such as avoiding macrophages and also neutrophils. The bacteria's innermost layer is known as a plasma membrane, and plasma membranes are also found in human cells, for example, in eukaryotes. And the structure is very similar in both bacteria and humans. The plasma membranes are semi-permeable membranes allowing transportation of substances in and out of the cells. And plasma membranes, as I mentioned, in bacteria are similar to those in humans in that bacteria, the plasma membrane is also a phospholipid bilayer and contains proteins as well within the membrane. The phospholipid bilayer is made up of molecules known as phospholipids. A phospholipid has a hydrophilic region, which means a region that loves water, hydro as in water, philic as in love, and also has a hydrophobic region, hydro as in water and phobic as in scared. And this characteristic of a phospholipid basically enables it to form the phospholipid bilayer. And so that's for the plasma membrane. Now, in between the capsule and plasma membrane, we have the cell wall. And this is a very complex semi-rigid structure. And the cell wall enables bacteria to be classified into two major groups: gram-positive bacteria or gram-negative bacteria. And there's a difference. First of all, they are grouped into gram-positive and gram-negative bacteria because when you dye them with different dyes, they have different colors. And the different colors are caused by the different cell composition of both these types of bacteria. So what is the difference in cell wall composition? Well, the gram-positive bacteria has a cell membrane with a structure above it. Let's just say here's the outside of the bacteria and here's the inside of the bacteria. So here we have the cell membrane, and the structure above it is known as a peptidoglycan and is composed of amino acids and sugars. The gram-negative bacteria also has a cell membrane, but it has a very thin peptidoglycan layer compared to the gram-positive. So gram-negative has a thin peptidoglycan layer, the gram-positive has a thick peptidoglycan layer. In addition to that, the gram-negative has an extra cell membrane above this thin peptidoglycan layer. The other notable difference between gram-positive and gram-negative bacteria is that the gram-positive bacteria has what's called a lipoteichoic acid, which enables it to bind to cell walls or be recognized by macrophages or phagocytes. The gram-negative bacteria, on the other hand, has a lot of lipid-like structures on the outer cell membrane, such as lipoproteins and also what's called lipid A. Lipid A are toxins, and they cause certain diseases and shock syndromes within the human body. So now, because of these different cell wall compositions, when we use dye, these bacteria, depending on what type of cell composition it has, will have a different color. The dyes used to stain a particular bacteria in successive order is crystal violet, and then we use iodine treatment, and then we decolorize it using alcohol, and then we use finally safranin. And essentially, I'll make a video hopefully on how this process works, but essentially the bacteria with a thick peptidoglycan layer and one cell membrane will have the crystal violet color, which is a bit purplish at the end, and the bacteria which has two cell membranes and a thin will have a pinkish red color. And so this is how we classify the bacteria. If it has a purple color after being dyed, it's a gram-positive. If it has a pinkish color after being dyed, it's a gram-negative. Hope this makes sense. So now that we know about the layers of the bacteria has, let's look at the cytoplasm of a bacteria. Cytoplasm contains water, enzymes, ribosomes, and circular DNA. And this is the fundamental difference between a bacteria and a eukaryote, or human, for example. So ribosomes are, as we know, are the site of protein synthesis, and they can be found all around the cytoplasm here. The DNA in bacteria is circular and not linear, such as the DNA in humans. But it should be noted that humans also have circular DNA in the mitochondria. But anyway, the main DNA that make up the bacteria are in a circular form, and they're all tangled up, and this is the genetic material. And we also have additional genetic material known as plasmids that are in the cytoplasm, and they're not part of the circular DNA. They're like a small circular genetic information around the cytoplasm. And plasmids, as I mentioned, have additional genetic information, and they could be incorporated in the circular DNA, the big one, or they could be transferred between two bacteria through conjugation, through the fimbriae or pili. So that's all on the bacterial structure. In the next video, we'll look at the bacteria's growth, reproduction, and also classifications.